rounds. We have two speakers today from Yale, two wonderful speakers. First, Jorge Galan, whom I'll introduce, and then Nicole Diesel, who Melinda Irwin will introduce. I've known Jorge since before he arrived at Yale. He's now the Lucille P. Markey Professor of Microbial Pathogenesis and chair of the department. In his lab studies bacterial infections. He's probably best known for his discovery and characterization of the type 3 secretion apparatus, which is this microscopic needle that injects proteins into cells. It's a fantastic story. And in recognition of this work, he's received numerous awards and been elected into the National Academy of Sciences. But more recently, he's gotten interested in the association between cancer and bacteria, and mechanistically, how does that work? And he's shown, for example, that some bacterial toxins can induce DNA damage and therefore are potential carcinogens. And so today we'll hear about this very exciting work. So Jorge, the floor is yours.
Thanks, Dan. Thank you very much for the introduction. And yeah, as Dan imply, microbes and cancer are a lot more intertwined than many would think. If we start with the obvious that 20% of cancers are caused by microbial infections. You know of all these examples of gastric cancer and Helicobacter, anogenital cancer, and HPV, and so on and so forth. So these are the known causes of microbial causes of cancer that amount to that 20%. But by all accounts, this is probably a gross underestimate in terms of the influence in the etiology of cancer of microbes. But microbes are intertwined with cancer in many other ways. For example, the study of microbial pathogens really have provided fundamental knowledge for the understanding of cancer. You, of course, are aware that oncogenes really were discovered through the study of chicken retrovirus. And today, the study of host-pathogen interactions have provided insight into cancer that are extremely important for the fundamental knowledge of this disease. Of course, infectious diseases as an entity are really a significant challenge in the context of cancer patients. Many of the drugs that we administer to treat cancer are, of course, have immunosuppressive power, and that increases the susceptibility of patients to infectious diseases, and it poses a challenge in many therapeutic settings. And of course, the elephant in the room, the resident microbiome, which in the last 5 years or so is quickly emerging as a major factor, both in terms of cancer etiology and cancer treatment. So what I'm trying to tell you here is something that it should be obvious, and that this is an extremely important aspect of cancer biology, and quite frankly, is one of the most exciting times to be involved in this research space. So that's perhaps the reason why maybe some of us were a bit disappointed when the cancer microbiology piece of the Cancer Center was interrupted. In fact, when I was invited to give this talk, I was invited as a member of the cancer microbiology group, which now doesn't exist. And although I completely understand why the leadership of the Cancer Center took this step, they had to tend to an impending cancer center grant that that obviously dictate many of these things. And it is clear that cancer microbiology or microbes really are not the so-called cancer establishment from which reviewers will be drawn to review the Cancer Center grant, are not really friendly to the concept of microbes and cancer for whatever reason. And even other considerations, other currencies that are used in the evaluation of these grants, such as, for example, NCI grants, people working with microbes that don't score high on that, because it would be a fundraising malpractice if you can send your grant to NIAID to send it to NCI, which is much less generous and certainly less friendly to these causes. So although I totally understand this, I think is an opportunity lost for leadership, particularly with the history of Yale, with the tremendous leadership, people like Dan DiManio or Charles Miller, in the space of oncovirus, for example. There is a lot of history here on this space. But life is life, and the cancer establishment is the cancer establishment. This is the same establishment that 10 years ago or 20 years ago were snotty related to cancer immunology. It were people like Lloyd Old, the late Lloyd Old, that was advocating for it. And he was really, honestly, looked down. I was a close collaborator of Lloyd, and he always complained. Of course, he was shielded by the Ludwig Cancer Center. He didn't have to worry about it. But anyway, I think it's more of a lost opportunity, but I totally understand why this decision was made. So enough venting, enough proselytizing. Let's get back to business here. And in order to put in context a little bit what I will tell you briefly is to consider the general mechanisms by which microbes induce cancer. And there are two types of mechanisms, if you will. The direct oncogenesis, and that is obvious when a virus introduces an oncogene in cells. This is, of course, the mechanisms behind HPV or EBV, for example, or when the integration event itself acts as an oncogene, because obviously the viruses integrate upstream of some gene that can drive sort of proliferation and growth. So that's direct oncogenesis. But arguably more common is the indirect oncogenesis. And this takes several forms. For example, the form in which viruses cause immunosuppression, and immunosuppression activates, for example, other tumor viruses. In the case of Kaposi sarcomas and HIV infection, this comes to mind. Or when viruses, viral infection, for example, triggers chromosomal instability or translocation, that eventually, that of course leads to cancer. And other aspects of the indirect oncogenesis, more related to what I'm going to be telling you today, is, for example, chronic inflammation, which is very well established to be linked to oncogenesis. The production of pro-inflammatory cytokines that have growth promoting abilities, combined with oxygen radicals that have a mutation or mutagenesis ability, leads to setting the stage for oncogenesis. Is the case, for example, with Helicobacter pylori and gastric cancer. And in addition, something that has been emerging over the last few years and that we sort of pioneer in this area is the fact that certain organisms really produce direct genotoxins that will drive the oncogenic event. So as I alluded, these last two are the ones more relevant to bacteria, which is the type of microbes that we study in the lab. So bacterial, in this context, bacterial colonization leads to both inflammation and genotoxin production and exposure of tissues to genotoxin, and therefore predisposing those tissues to cancer. What I'm going to be telling you today is the paradigm of two organisms that we study in the lab. Both of them, these organisms are Salmonella and Campylobacter jejuni. Both of them have been very strongly epidemiologically associated with the development of cancer. Campylobacter jejuni associated with an intestinal lymphoma, and while Salmonella Typhi, one of the Salmonella that we study in the lab, is really a major cause of gallbladder cancer. And gallbladder cancer in endemic areas is actually one of the main cancers that affect those individuals. And infections with Salmonella Typhi are associated with a 200-fold risk of hepatobiliary carcinoma and gallbladder cancer. So these are important causes of cancer, which incidentally are not in that 20% statistic that I told you about. Now, in the case of Campylobacter, what we discovered that was sort of central to understand how these organisms linked to oncogenesis is the characterization of a toxin that we did almost 2 decades ago, actually scary, more than 2 decades ago. And that is a toxin that caught our attention because of what you see here in these images. These are cells that are intoxicated. You see them very much expanded with a large nuclei. in comparison to a control cell at the same
Excuse me,
magnitude.
Jorge, your
Yes,
slide did not advance.
Appa. You mean in what they haven't
You're still on the
advanced?
first slide.
Oh gosh, that's not good. That's, can you see them this way?
Now, yeah, I can see those.
Okay, when I do it that way, because
Okay.
you know, unfortunate, because whatever, it would have been easier, but thank you for letting me know. Okay, so anyway, so here it is, okay, so this image is showing you the cells that have been intoxicated with this toxin, showing this unusual morphology in comparison with control cells. And the reason these cells have that morphology is because the cells are stuck on the G2M phase of the cell cycle. And we found that the reason for that is that this toxin that we had discovered it has a genotox, has a DNA damaging capacity. This is a toxin, a typical toxin of we call AB toxins, have two types of parts, if you will. The B part, which is what targets the payload to a particular cell, and the payload part, the nuclease, is an endonuclease. It's actually an unusual endonuclease in the sense that primary amino acid sequence would not overtly tell you that this is a nuclease, but when you look at the atomic structure, you can make out the catalytic site. So this is a typical case of a genotoxin that is responsible for driving the cancer development. And in fact, just recently, this has been formally demonstrated in an animal model that that this toxin is responsible for Campylobacter jejuni's ability to promote cancer. So it's kind of awkward to have to advance it like this, but I will go ahead. So the second example is Salmonella Typhi. And I need to tell you just the basic about Salmonella Typhi to put things in context again. And that is that Salmonella Typhi is an exclusive pathogen of humans. It causes typhoid fever, one of those historical diseases, if you will. But important for oncogenesis is the fact that those that survive the disease, many of them go on to persistently harbor the organism within the gallbladder. And that is where the rubber meets the road. And that is the reason why those individuals that are harboring Salmonella Typhi in the endemic areas are prone to develop gallbladder cancer. And in the case of Salmonella, the paradigm is slightly different than the paradigm in Campylobacter jejuni, but it shares, remarkably, it shares more than what would expect. You need to think in terms of evolution that Campylobacter and Salmonella they couldn't be more far apart. One is an epsilon bacteria, the other is a gamma protobacteria. It's like absolutely no evolutionary connection. And yet, what is remarkable is that we discover a toxin in Salmonella Typhi that we call typhoid toxin that also has the ability to induce DNA damage, as shown here in this image. And when we characterize this toxin, we were surprised to see that the active subunit of this toxin was virtually identical to the active subunit of the completely unrelated toxin carried by Campylobacter jejuni. So this is really a remarkable piece of evolution. This is one of those head-turning toxins that actually evolution put it together by fusing two toxins. One that some of you may be familiar, called pertussis toxin, is what makes you what is central for the pathogenesis of whooping cough. And then this other toxin that I described earlier, the cytolethal distended toxin. So this lower part of the toxin comes from pertussis toxin, and this upper part of the toxin comes from cytolethal distended toxin. So evolution hooked them together to make this head-turning toxin that Salmonella Typhi encodes, and that it is responsible for the genotoxicity of this organism. And this is an unusual toxin in many different ways that I don't have time to go into. But one of the remarkable ways in which this toxin is unique is that it is adaptation to the human host. And what do I mean by that? Well, the receptor for this toxin, or the receptors, I should say, we discovered are two proteins, podocalycin 1 in epithelial cells and CD45 in immune cells. But what is important here is what does the toxin see on these glycoproteins, and is the glycan part. And through glycan arrays and other types of studies to address these kinds of questions, we discovered that what typhoid toxin likes, if you will, is glycans terminated in acetylnuraminic acid and hooked to galactose and to glucose or acetylglucosamine in this particular fashion. And why is this relevant? Well, this is important because you may not know this, or many of you may, is that we humans are actually rather unusual mammals in many ways. And one of the ways in which we are unusual is in our glycosylation pattern. All our sialoglycans are terminated in acetylnuraminic acid, but all other mammals, in fact, glycans are terminated in glycolyl-nuraminic acid. And the reason is that the enzyme that is responsible for, can you see this slide? And maybe, well, whatever, I continue this way. The enzyme that is responsible for putting this oxygen here is mutated in humans. We have a pseudogene there, and therefore we are unique in that fashion. And typhoid toxin combined glycans exclusively terminated in acetylnuraminic acid. In fact, if you just change one oxidant in any of these glycans that typhoid toxin likes, and you array them in an array, in a glycan array, typhoid toxin does not bind at all. So it has the ability to distinguish just one atom of oxygen. Remarkable piece of evolution that makes it able to target human cells and have that oncogenic effect. But in addition to having a genotoxin, Salmonella typhi, it actually has the 2-for-1. In other words, it's also the chronic inflammation part that plays a role in the oncogenesis of Salmonella typhi. And so Salmonella typhi causes chronic inflammation of the gallbladder. That chronic inflammation, as is well known, leads to the development of cancer, or contributes to the development of cancer. The paradigm here is well known, where, as I said, the production of growth-promoting cytokines combined with or radical oxygens that are mutagenic eventually leads to the development of cancer, angiogenesis, growth stimulation, and so on and so forth. So the issue is, how does Salmonella trigger inflammation? Now, on the surface, this could be a rather simple story. And you may know that we are endowed with innate immune receptors, famously put into the scientific space by the late Charlie Janeway here at Yale. And these innate immune receptors have the capacity to recognize bacterial products, lipopolysaccharide, peptidoglycan, flagella, you name it. Many bacterial products can be detected by these innate immune receptors that essentially coordinate an inflammatory response. And that inflammatory response eventually leads to pathogen rejection and the acquired immune response. So this is central to the way we hosts, not just humans, but all mammals, defend against microbial pathogens. Now, it turns out that then from this framework, it would be very simple to think that Salmonella triggers inflammation simply because it has plenty of LPS, it has plenty of these products, and it's the detection of the host that drives inflammation. In other words, this would be like a host-centric view. But work that we have done in our lab for the last 15 years or so has completely turned around this paradigm and discovered that that's actually incorrect in the case of Salmonella. That Salmonella really has a specific adaptations evolved by Salmonella to trigger inflammation. So this is a pathogen-driven process, not a host-driven process. And the reason is very simple, or not so simple. Salmonella, like many other microbes, when they encounter a mucosal site, being an intestine or being the gallbladder mucosa, they need to compete with resident microbiota, who has a foothold on that tissue and really put up a good fight. This is actually one of our main barriers against bacterial pathogens, particularly in the intestine, is the resident microbiota. The inflammatory response causes a profound dysbiosis, which is essential for Salmonella to be able to colonize and replicate. Not only that, inflammation makes nutrients available, electron acceptors, camphor sources, that otherwise would not be available in the uninflamed tissue, and that drives the replication of the luminal population of Salmonella. So it's the inflammatory response that causes, even though Salmonella is an intracellular pathogen, the bulk of the bacterial replication comes from this luminal population that is fed from the inflammatory response triggered by this bacteria here. So a nice division of labor. Now, how does Salmonella manage to trigger an inflammatory response at mucosal sites, which actually is pretty difficult because mucosal sites are subject to very stringent negative regulation of its innate immune receptors, precisely to prevent this microbiota that in theory can also stimulate innate immune receptors to trigger an inflammatory response, and for all of us to be walking with IBD or Crohn's disease. To avoid that, there are very precise mechanisms to keep those innate immune receptors in check. And Salmonella has to trigger inflammation in that environment. How does it do it? Well, it does it through this amazing machine that Dan alluded earlier, that we discovered more than 2 decades ago. It's an amazing sort of a bacterial injection device, if you will, that injects bacterially encoded proteins that have the capacity to modulate many signal transduction pathways. And it's the ability to stimulate those signal transduction pathways and modulate cellular process for the benefit of the pathogen. Specifically relevant to inflammation are 3 of these effector proteins that activate Rho family GTPases by either being exchange factors of these Rho family GTPases or phosphinoinositide phosphatase in the case of this particular effector that would activate endogenous exchange factors and activate that. And that leads to the activation of CDC42, and then the activation of CDC42 leads to transcriptional responses that really are pro-inflammatory and really look like innate immune responses. This was a bit puzzling for a number of years because CDC42 had never been linked to innate immune responses until very recently, where we sort of cracked this little puzzle, and we discovered that the activation of CDC42 by Salmonella leads to the formation of a non-canonical signaling complex made by PAC1, a target of CDC42, and these other components, TRAP6, TAC1, TAP1, TAP2, that leads to the inflammatory response. And what explains the whole thing is that this signaling complex is identical to the signaling complex that is tripped by innate immune receptors. So what Salmonella in essence is doing is going down the signaling pathway so as to avoid the negative regulatory system and trigger essentially an innate immune response, but by non-canonical methods. So it uses other type of mechanisms similar to this, going downstream of canonical signaling pathways. But since my time is up, I'm just going to put up sort of a summary of this and sort of to give you a flavor of how these effector proteins can go down different signaling pathways, intersect with signaling pathways. For example, this effector can activate the RIG-I and MDI5 nucleotide sensing pathway, but without the need of nucleotides. It just simply activates RIG-I by interfacing with TRIM56 and TRIM65, 2 regulators of this pathway. It ubiquitinates them, activates them, and trips this signaling pathway without the need of the agonist of those receptors. And the same applies to another effector here, that it actually inhibits an anti-inflammatory pathway. But since I don't have time, I had to skip it. So I hope that you got a sense of the sophistication by which bacterial pathogens manipulate cells in ways that benefit them but doesn't benefit us, and that through the production of genotoxins or inflammation, it leads to the predisposition to cancer. And then finally, last but not least, people that were involved in this work, obviously I talked to work that was done a number of years ago as well, but for the inflammation pathway, Hui and Yana were involved, and Juan, who did all the SOP-D work, and Maria, of course, has made the pioneering discoveries of the genotoxin in Campylobacter jejuni and set in motion all this work. And thank you very much. And with that, I'm gonna stop sharing if I can. And They need
Okay, well, thank you very much, Jorge. It's very interesting, very exciting work.
I don't know how to, well, I should know how to stop sharing, but oh, there you go. You helped me on that one.
There you
Yeah.
go. Okay.
Yeah.
So if people have questions, you can type them into the chat, or Renee, is there a way to unmute them?
We can if you'd like, sure.
I'll ask a quick question. I was
Yeah.
very struck by by what appears to be the convergent evolution of these two nucleases.
Yeah.
It's really very striking. What is the advantage of the bacteria to induce cell cycle arrest?
Well, cell cycle arrest actually is also pro-inflammatory. So probably one of the main drivers is the pro-inflammatory response. And also, in the case of Salmonella typhi, is using this activity to target immune cells. So obviously, if you're a virus and you know a thing or two about them, and you integrate your genome in the host, you're free. That's the way you can persist as long as you want. If you're Salmonella typhi, you have 4,761. Those are the number of open reading frames. Potential antigens you need to hide. You can't do that, right? So the way Salmonella typhi does it is by creating a sort of a immunological suppression around the site where it colonizes. And this toxin is central for that, for the persistent infection by targeting immune cells. And in the case of Campylobacter, of course, inflammation is central for the bug. And this pro-inflammatory aspect of DNA damage is probably what evolutionary selected for these genotoxins. And in the process, we got clipped.
Thank you. Are there other questions for Jorge?
All right. Well, thank you very much.
I think Dan froze. Oh, you were frozen, Dan.
Oh, I'm sorry. Well,
You were frozen.
I just introduced you, Melinda.
Okay, great. Don't we love the advances in technology? Thank you, Dr. Galan. That was fabulous. And I am now delighted to introduce Dr. Nicole Diesel to present her research on environmental carcinogens and thyroid cancer. Dr. Diesel is an associate professor of environmental health in the Yale School of Public Health, and she received her PhD and her master's in industrial hygiene from the Johns Hopkins School of Public Health. Her environmental exposure assessment strategies aim to reduce exposure misclassification for epidemiologic studies and an advanced understanding of the relationship between exposure to environmental chemicals and the risk of cancer and other adverse health outcomes. She serves as the PI of a study funded by the American Cancer Society and investigating exposure to flame retardants, pesticides, and other persistent pollutants in thyroid cancer risk. And of note, she is the winner of the Yale Cancer Center 2020 Research Prize in Population Science for her research on this topic. So we're delighted for your presentation, Dr. Diesel. Take it away.
Okay, thank you so much, Melinda, for that generous introduction. I'm very pleased to be here to share some of my recent work with you. I really enjoyed Dr. Galan's presentation looking at exposure to microbial pathogens and the associated toxins. And I'll be switching gears a little bit to look at work looking at the epidemiology of exposures to chemical toxins and thyroid cancer risk. Okay, so I've advanced my slide and someone can let me know if there's any issue there. I first wanted to take a moment just to tell you about the motivation of my research and the research I'll be presenting today and talk about environmental risk factors for cancer. We know that a third of cancers are attributable to modifiable factors. We often think of things related to diet, alcohol, tobacco, these so-called lifestyle factors, but this also includes infections, which we just heard about, as well as pollution. So how much of cancer cases can we attribute to this pollution? Well, Deziel and Pito in their landmark study estimated about 7% of cancer deaths could could be attributable to occupation, pollution, and industrial products. And many experts agree that this percentage is likely grossly underestimated due to the extremely limited data on the commercial chemicals that we encounter in our day-to-day lives. So in the United States, there are 80,000 chemicals that are licensed for commercial use. And of those, only 200, so not even a percentage of them, have been screened adequately for carcinogenicity. And every time I share this statistic, I find it really striking. Another reason why this is so important is that, many of these exposures are outside individual control. These are things in air pollution, our water supply, the food supply, our workplaces. So we really rely on the government to protect us from exposure to these potentially harmful chemicals. And our regulatory system really is quite inadequate to serve this purpose. The way it's structured, chemicals have to be proven harmful rather than proven safe at the outset. So it normally requires researchers like myself and others in my field to study chemicals, for decades before we acquire enough evidence to demonstrate harm for a particular chemical. And also importantly, we know these exposures are not distributed equitably across populations, and that populations experiencing other social disadvantages are often disproportionately exposed to certain pollutants. And some of these points are highlighted in a forthcoming book chapter that I worked on with Dr. Irwin, edited by Charlie Fuchs. So turning to the specific research I want to talk about today, which is related to thyroid cancer. Thyroid cancer is one of the fastest growing malignancies. It has nearly tripled over the past few decades, as you can see in these graphs of SEER cancer incidence data. You can also note by looking at the y-axis that females have 3 times the incidence compared to males. And thyroid cancer, has a very good prognosis. It's more than 90% survival after 20 years. However, survivors really face many physical, psychological, and financial challenges with the prolonged treatment, increased surveillance, risk of second primary cancer, and other quality of life issues. So this increase is certainly due, at least in part, to improvements and changes to diagnostic techniques, imaging techniques, and fine needle biopsies. So there's some debate about what proportion can be attributed to this increased diagnostic scrutiny, but many analyses suggest that about half of this trend can be linked to these diagnostic changes, leaving half for environmental or lifestyle factors. So we've hypothesized that increasing exposure to thyroid hormone disrupting environmental chemicals, such as these polybrominated diphenyl ether flame retardants, or PBDEs, may be partially driving this increasing trend. So I'll just talk a little bit more about these PBDEs. There are actually a lot of thyroid hormone disrupting environmental chemicals in use and in the environment. These flame retardants were widely added to many different products. The polyurethane foam in mattresses and couch cushions and vehicle seats, including baby car seats. They were also added to electronics like phones, cell phones, televisions, computers. And the reason they were added was to meet flammability standards such that if these products, caught fire, they would burn more slowly. which is a good thing from a public health perspective. However, these chemicals, once added to these products, did not stay bound in the matrices as indicated by their manufacturers, and instead have migrated out into our homes, home environments, cars, workplaces, etc. So due to this widespread use, as well as disposal and improper disposal of these chemicals, they are ubiquitous, and more than 90% of the population here in the US and globally are exposed to these chemicals. Um, and this also, they are extremely persistent. Once they get into our homes or our bodies, they do not degrade very easily. So they stick around for years and decades. So due to concerns about this persistence and potential toxicities, these particular group of chemicals, the PBDEs, were phase outs were initiated over the last, in the past decade. However, exposures do continue for the reasons I described, their persistence, their presence in products made before then, as well as their, presence in the food supply and elsewhere. One other group of chemicals that I'll be talking about today are the polychlorinated biphenyls, or PCBs. These were also used widely in electrical equipment, hydraulic machinery, construction materials. These were banned in 1979. So again, you might say, why are we even studying these now? Well, they're still around, and they're still around in our bodies, our bloodstreams, in the environment. And in fact, here in Connecticut, there's been some renewed concern about these legacy chemicals. They were commonly used in buildings, including schools constructed in the 1950s to 1970s. And many of these schools now are in need of repairs and renovations. And there have been some notable schools in Connecticut that have had PCB levels exceeding safe levels, closures of schools, insufficient funds to do a proper and safe remodeling or renovations. And again, often these are in kind of environmental justice communities. And also another reason to study these legacy chemicals is as they get phased out, new chemicals come to take their place, and many of those are also have similar properties. And so understanding these may help us inform, greener chemistry or future regulations of other chemicals. So we hypothesize that these PBDEs could be contributing to that increasing trend in thyroid cancer. Here is a graph showing increasing exposure over a similar time period where we saw thyroid cancer cases start to go up. So these are measurements taken from blood samples from a blood bank in the US. And you can see about a doubling every 5 years of these particular chemicals. So looking at more recent data, because as I mentioned, these have been somewhat phased out. You can see that while levels have come down since some of those earlier years, but then they really have somewhat plateaued or are, some particular congeners of these in this family of chemicals are still increasing slightly. And we see similar trends with the PCBs, that levels have come down since they were banned, but then they reach this plateau in the population because of their persistence. And then as new chemicals come on, we may be introduced to those on top of these exposures. Okay, and so why do we think these may be linked to thyroid cancer? These chemicals are established thyroid hormone disruptors. They're endocrine disruptors. And over here, I have an image of our thyroid hormone thyroxine, and then the PCBs and the PBDEs and their general chemical structure. So you can see immediately how structurally similar these chemicals are to thyroxine. We've got the two aromatic rings, and then while thyroxine has iodine, these chemicals have other halogens. They have either chlorines or bromines. And just to further illustrate, when, for example, this particular PBDE gets metabolized, it gets this hydroxyl group added, and now it even more closely resembles thyroxine. So in vitro studies have shown that these chemicals can competitively bind with thyroid transport proteins and result in reduced circulation of thyroid hormones, which could then result in dysregulation of the transport and signaling pathways, potentially leading to overproduction of hormones to compensate proliferation in the thyroid and potentially neoplasia. So as you know, the hypothalamus pituitary thyroid axis is this very well choreographed system. So these perturbations or dysregulation of these systems can perhaps trigger some proliferation of the thyroid and potentially lead to thyroid cancer. I'm showing this slide to also illustrate that some chemicals have some additional hypothesized mechanisms, such as some have been shown to be capable to directly bind to DNA, leading to mutations, and could potentially lead to carcinogenesis that way. So before we launched our study, there had only been 2 other studies examining this hypothesis, this idea that these PBDEs could be linked to thyroid cancer. So the first 2 rows of this table are the 2 prior epidemiologic studies, and the 3rd row is the study that I led. A couple things to point out here is that the studies that came before us had, moderate study populations, and ours was larger. We also looked at more chemicals, and importantly, we looked at single and multi-pollutant models. So what I mean by that is that in the real world, we're not exposed to one chemical at a time. We're typically exposed to groups of chemicals or mixtures of chemicals. And traditional environmental epidemiologic studies have looked at chemicals just one at a time. And in my work, we're looking at these so-called mixtures using different modeling techniques to look at the joint effect of exposures to multiple chemicals. Similarly, for the Pcbs, the other chemicals, there were also only 2 previous studies before we did ours. Also, the other studies were a bit smaller, and we also looked at mixture modeling. One shortcoming I wanted to point out, actually, in reference to at least one of the other studies, is that our study was a case control study, so we did collect serum samples to do our environmental chemical measurements after diagnosis, whereas one other study collected pre-diagnosis samples. We think, so we're using this post-diagnosis sample to try to capture exposures in the past. While it's not optimal, we think this is actually a pretty strong and reasonable assumption, because we know that these chemicals have half-lives of years and decades, and may well reflect past use and past exposure. Okay, so just a few more details about the study we conducted. I led a study within a study, and that larger study was the Connecticut Thyroid Cancer Case Control Study, which was led by Yawei Chong, who was our former colleague here at the Yale Cancer Center. We focused on women because they have that 3 times higher risk I showed earlier. About 90% of our cases were white, so we focused on the white population because our numbers were really small for looking at other demographics. I do think now this is something I would like to follow up on more in another population. We focused on papillary thyroid cancer, because that's about 85% of the new cases are papillary. We also collected very detailed information about demographics, lifestyle, diet, many other risk factors so that we could control for other factors. We collected the blood sample at the time of the interview and then measured the participants' blood samples for 11 different PBDEs and 32 Pcbs, which this analysis also gave us some pesticides like DDT, which are also structurally similar to these classes of chemicals. And again, we looked at both pollutants one at a time, like most studies do, and then multi-pollutant models. Okay, so here's some results from our study population. These are factors that differed between cases and controls. So our cases had a lower educational attainment compared to controls. They did have a history of benign thyroid disease. That's a strong risk factor for thyroid cancer. Alcohol consumption was actually higher, lower in cases. This is one of the few cancers where alcohol actually has been consistently shown to have a protective effect. And cases also had a history of thyroid cancer and higher BMI, which has been shown now in several studies indicating another possible risk factor that can also be interrelated to like endocrine disrupting chemicals and also. Okay, so here are some results from our single pollutant models where we looked at one chemical at a time. These are increasing categories of exposure within each of these different pollutants. Odds ratio of 1 means no effect, and anything above 1 would indicate an association with thyroid cancer. So you can see here that there's really not much going on with these individual models. If anything, the odds of thyroid cancer seem to be lower in those who are exposed compared to the reference group of low exposure. So medium and high groups are not at an elevated odds of thyroid cancer, except for this one chemical, this Pbb 153. So when we move to our multi-pollutant models, the results are also somewhat null. We have 2 multi-pollutant models here using Bayesian modeling, which I won't get into here, but here you can see that the picture is still pretty null, except for in this case we have this a different chemical, PBDE 100, was associated with elevated odds of thyroid cancer using both those models. And then finally, we did this one more mixture type approach, a principal components analysis. And in this work, we found that people who had this combination of exposures, which was higher PBDE 153 and lower PBDE 209, had elevated odds of thyroid cancer. So then moving on to the Pcbs, for this one, because we had so many, we had 32 different chemicals. I'm just going to present some summed groups of structurally similar Pcbs, which was another approach we used to look at groups of chemicals. So in this model, we're just looking at one group at a time. The odds ratios are hovering around 1. So again, pretty null findings. We're not seeing a link between exposure and thyroid cancer. However, so the most intriguing part of this study was when we took a closer look at the groups of people who were exposed who were younger during peak production, who were born during peak production of PCBs, so presumably would have their highest exposure in very early life. They consistently had higher odds of thyroid cancer, including this group of PCBs that were particularly structurally similar to thyroid hormones. So this was quite intriguing to me, suggesting maybe the timing of exposure could be important. Okay. So just to summarize some of the key takeaways from both of these studies, some strengths were that we looked at this larger population and incorporated these different models to account for co-exposures to multiple pollutants. The results were generally null. We did see a few chemicals here and there associated with elevated odds of thyroid cancer. Particularly, this was more consistent when we looked at the group of women who were born during peak production of Pcbs. However, we only have 3 studies now for each of these groups of chemicals, so there's really insufficient evidence to rule them out. And I think there can be some improvements to the study design to try to look at this more carefully. I think looking at early life would be important, and using a prospective design where we could have samples collected pre-diagnosis could help, really try to nail down if anything is going on here. And then finally, I just want to talk about where I'm taking this work. I've now expanded this work in adults to looking at children. So with my collaborator, Xiaomei Ma, also very active in the Cancer Center, we are looking at environmental exposures and pediatric thyroid cancer. So here's some incidence data on pediatric thyroid cancer. It has also been increasing over time, and children are less likely to be targeted for increased screening and diagnosis and imaging. So these trends are concerning. So we have some projects underway to try to look at environmental exposures in this more vulnerable population. And with that, I would like to acknowledge all my wonderful collaborators and my funding from American Cancer Society, as well as the Yale Cancer Center, for getting me started in this line of research. Thank you.
Thank you, Dr. Diesel. That was fantastic. A little alarming, I would say, when we start to think about how many, carcinogens exist in our environment, and the fact that I'm intrigued by the policy statement that we have to show harm before anything can be done and have these substances banned. Do you think there's any potential to being able to reverse that or change that policy, or will it take years and more data to do so?
Yeah, so a lot of the chemicals were kind of grandfathered in when we established the Environmental Protection Agency in 1970. There is a new act that is supposed to reverse this burden of proof, but I don't think it's going to be retroactive. So I am encouraged too with the current administration that we may start to move towards a different model. Also in Europe, they have stronger precautionary policies where if there's a safer alternative, you have to use it, and not to wait until we prove something with certainty to take some sort of action.
Great. So there's a couple of chat questions. I'll just quickly read them. Herta Chow asked about any data on Agent Orange and thyroid cancer.
Oh, yeah. So dioxins is one of the constituents of Agent Orange. Well, Agent Orange had, these defoliant chemicals, so various herbicides, and then dioxins. That's a great question, because they're also very structurally similar to the other chemicals I presented. But I'd have to, and some of the chemicals we looked at, some of the groups were dioxin-like. I'm not sure of any specific studies coming to mind, but it seems likely that it may follow a similar, well, they're structurally similar to the other chemicals.
Okay, great. And then Jeffrey Townsend has a question. Where you do see elevated odds of thyroid cancer, do you have any evidence discriminating between the two hypotheses of effects that occur due to hormone disruption compared to effects that might be arising due to induction of carcinogenic mutation? Do you see ways to do this?
Yeah, that's a great question. I don't really do this type of mechanistic work. I know the the thyroid hormone disruption hypothesis, there's been a lot more mechanistic work in that area. But I'd be open to suggestions for how this could hopefully maybe inform some mechanistic studies. I think it would be, I really think that this study can inform mechanistic work and vice versa.
Yeah, that's a great point. And then Ashita had mentioned in regards to the policy of showing harm that Europe does the reverse of the US. Do they see as a different trend?
Actually, thyroid cancer cases are going up globally, and the trends in exposure are consistent. Actually, they were first observed in Sweden, where they have breast milk banks, and they saw these flame retardants going up in human milk samples, which caused a lot of alarm. At that time, we didn't know if they were carcinogenic or not, but given that babies were going to be exposed to these chemicals, that sort of triggered this whole area of research on these flame retardants and other chemicals.
Great. And then I just have one final question. The email that we all received last week about benzene being in some of the hand sanitizers for our COVID-19 protection. Do you have any thoughts or comments on that?
Yeah, I read some of the materials. I think benzene, is a known human carcinogen. However, it is present in many sources. So I would really want to understand how risky this is. I think we need to know how much benzene and how does that compare to like putting gasoline in your car, or walking near the roadway. So it didn't raise too many alarms yet, but I feel like I need more data to be able to reach that conclusion. And I mean, so Yale Health, Environmental Safety and Health said, like, let's be precautionary. Let's just get rid of these sanitizers. Let's take an action before we have all the answers. So I think that's a, very sensible approach.
Okay, great. Well, thank you so much. Dan, do you have any other closing comments?
I'll just thank both our speakers today. 2 terrific talks. I learned a lot. Thank you.
Yes, thank you. Have a great day.
Bye.